The need for drugs have been rising for decades. Most conventional drugs work by eliminating essential proteins. These solutions are helpful in healthcare, yet their functions are limited in scope. In the fight against disease, researchers have developed a new weapon capable of singling out enemy cells.
Enter the antibody-drug conjugate. Commonly referred to as ADCs, these molecules are able to lock onto and destroy threats to our body's well-being. So what is an antibody-drug conjugate? ADCs are complex molecules, thousands of times larger than the common small molecule drug, such as aspirin. They are composed of a specific antibody head connected to a cytotoxic payload. Antibodies are Y-shaped proteins created by our immune system to target cellular invaders, known as antigens. Antibodies attach to receptors on antigens and are able to mark the cell for destruction. By utilizing antibodies, ADCs are highly selective, binding only to the receptors of matching antigens. These chemical warheads are connected to the antibody through stable linkers, which are designed to stay together throughout the drug's journey. Upon binding with the cell's receptor, the ADC is brought inside through endocytosis, forming a vesicle to surround the drug. As the vesicle moves into the cytosol, lysosomes bind to and degrade the ADC, releasing cytotoxic agents into the cell. This payload can interfere with organelle function and effectively kill the cell. As ADCs are designed to be released only upon degradation, the payload can be extremely toxic. Their mode of action should not endanger other cells, thus reducing collateral cell damage. ADCs can be used to find cancer, as the antibodies could be made to target only mutated cell receptors. The antibodies themselves could also exhibit anti-cancer functions, working to promote apoptosis or stop cell signaling. You might be wondering then, why use ADCs when we could just poison cancer cells or target them with antibodies? Most cytotoxic drugs are potent and pose a large risk to the user. Simply injecting such toxic materials into the body would not only kill cancer cells, but could also terminate the patient. In 2019 alone, 3 ADCs were approved by the FDA for usage, accounting for almost 50% of all ADCs approved. This is a great indication for the future of drug design and even cancer research, signaling a new era of highly specific drugs.